and welcome to Miss Estruch Biology, and in this video I'm going to be going through everything you need to know for CIE topic 12, energy and respiration. Now if you do want even more help knowing the exact theory, key terms and examiner's tip, then don't forget to check out my A-level notes that covers all of the theory, and that is linked below for you. So topic 12, let's get into it, and it's covering energy and respiration. So first of all organisms require energy, and that is for metabolic processes such as active transport, where you're moving substances across a membrane, or other types of movement such as using a cilia or flagella or muscle contractions, and also for building polymers, so protein synthesis, where amino acids are then converted and joined together to make proteins. That requires energy, as well as DNA replication, joining together the DNA nucleotides. And energy can't be created and it also can't be destroyed, and instead organisms use energy that is coming from bonds. And the opposite of that is then making bonds to create molecules like polymers. That requires energy, but breaking them releases energy. So photosynthesis is an example, and this converts energy from the sun into actually creating bonds. Whereas respiration, this involves breaking bonds such as glucose, which is the respiratory substrate used to ultimately create ATP. And that ATP can then be hydrolyzed into ADP and PI, and when that bond is broken between the inorganic phosphates, that is what releases the small manageable amounts of energy which can be used in metabolic processes. So let's take a closer look at the structure of ATP. Now it's known as a nucleotide derivative because it's got very similar components to a nucleotide. It has this pentose sugar, it has a nitrogenous base. The key difference is instead of one inorganic phosphate group, it has 3 inorganic phosphate groups. And you need to know then that it is an immediate energy source for biological processes. Metabolic reactions in cells have to have a constant supply of this ATP, so they have the energy for reactions. And it's the bonds between these 3 inorganic phosphates which, when they are broken, the energy is released, and that's used for chemical reactions within the cells. So this structures then, we briefly touched on it because we said it contains a nitrogenous base. In ATP, that nitrogenous base is always adenine, and nitrogenous means it contains nitrogen, which we can see here inside of those rings. The pentose sugar is always ribose, and then it has 3 inorganic phosphates. So we can see here those bases, or that base adenine, the ribose and the 3 phosphate groups. Sometimes you might see it called adenosine triphosphate. Adenosine is the term for the adenine and ribose together. So it's adenosine with 3 phosphates. That's why it's adenosine triphosphate, or ATP. And the level of detail that you need to know it in is actually here. You don't need to know all of the atoms within it. You just need to know that you have the nitrogenous base bonded to the pentose sugar, which is ribose, bonded to these 3 inorganic phosphates. And the phosphate groups are described as being inorganic because they don't contain any carbon atoms. And for this reason, in chemical reactions, the symbol used to represent the inorganic phosphate is Pi, meaning inorganic phosphate. So a bit more about ATP then. And this time we've looked at the structure. Now we're going to look at the creation and hydrolysis of it. So ATP is made during respiration from ADP, which is adenosine diphosphate, by the addition of an extra inorganic phosphate. And that's a condensation reaction using the enzyme ATP synthase. And that's what we can see here, ADP plus PI, that results in the production of ATP plus water. And it's a condensation reaction, so that's why we've got plus water. Water's been removed. This is a reversible reaction. ATP can be hydrolyzed back into ADP plus PI. It's a hydrolysis reaction, so water is added, and with the help of the enzyme ATP hydrolase catalyzing this reaction, it will then split or hydrolyze ATP into ADP plus PI, and that's when energy is released, when the bond between those inorganic phosphates is broken. So this is done immediately. You don't actually store ATP. So that's why ATP is known as an immediate energy source. You also get this energy very rapidly because you only have to hydrolyze or break one bond to release that energy. And as well as releasing energy to the surroundings, ATP can actually transfer energy to other compounds. Because if you add that phosphate onto a different compound, that compound will then gain the energy. And that's what phosphorylation is. It's when you add an inorganic phosphate group onto a molecule so it becomes more reactive or it gains energy. So technically, this is actually a phosphorylation reaction because ADP is gaining a phosphate group to become more reactive or to have more energy in the form of ATP. And we'll also see this happening in the first stage of aerobic respiration because glucose gets phosphorylated as well. So energy values of respiratory substrates. Near the start of this video, I said that glucose is a respiratory substrate, but you can actually use other carbohydrates and lipids and proteins if there aren't other carbohydrates available. And all of these can provide energy to be used in respiration, but they all have slightly different energy values per gram, and that's due to the variations in the chemical structures and metabolic pathways. And that's what I've got summarized here on this table. So you might want to pause at this point and just copy this information. So we've got carbohydrates, lipids, proteins, glycogen, or if it's in a plant, it'd be starch. Glycogen's only for animals, and then alcohol. So we've got the energy yield per gram. So we can see you get the most energy from lipids, then alcohol, and then carbohydrates, proteins. Glycogen and starch are actually carbohydrates. This is referring more to sugars. And you can see the metabolic pathways and then the conditions of use. So that's a good one just to pause, copy down, or turn into flashcards if you want. So the respiratory equation then, or RQ, of each respiratory substrate can be measured using a respirometer, which is one of the practicals that you need to be familiar with. And you divide the volume of carbon dioxide produced by the volume of oxygen used. So that would be your formula, the carbon dioxide produced divided by oxygen consumed. And then you can use that to compare different substrates as well. So now we're going to move on to 12.2, which is respiration, both aerobic and anaerobic respiration. Starting with aerobic respiration, there are 4 key stages: glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation. You need to know where all 4 of those stages happen within the cell. Glycolysis happens in the cytoplasm, but the other 3 stages all happen inside of the mitochondria. The link reaction and the Krebs cycle are within the mitochondrial matrix, which is shown in blue on this diagram, and it's the fluid-filled center. Oxidative phosphorylation, the final stage, happens on the cristae, which is the name for this folded inner mitochondrial membrane shown in yellow. So let's start with glycolysis and the first stage. And this stage is where you are producing a small amount of ATP, but you also use a small amount of ATP. We said already it happens in the cytoplasm, and this stage doesn't require any oxygen, and therefore it occurs in both anaerobic and aerobic respiration. And there are 3 key steps that you need to know. The first step is substrate level phosphorylation, and this is when the glucose, which is the respiratory substrate, has 2 phosphate groups added to it from 2 ATP molecules. And that makes it a far more reactive molecule. And that's then what happens in the next stage, that phosphorylation. Once you've added those 2 phosphate groups to glucose, it creates fructose, 1,6-bisphosphate, which is a 6-carbon compound still, which is like glucose, 6-carbon compound. But because it's gained that energy from the joining or the bonding of those 2 phosphate groups, it makes it unstable, and therefore it splits immediately into 2 3-carbon compounds known as triphosphate or TP. Lastly then, those TP molecules, both of them, are oxidized, and they're oxidized by removing a hydrogen. And the reason removing a hydrogen is oxidation is a hydrogen atom is 1 proton and 1 electron. So oxidation is the loss of electrons. If you remove a hydrogen atom, you are removing an electron. So it's oxidized by the removal of a hydrogen atom, and that will then form 2 pyruvate molecules. Now also in this oxidation, we're going to create 2 reduced NAD. NAD is a coenzyme, which is a molecule that can pick up and transfer hydrogen from one part of the reaction to another in respiration. And we have NAD gaining the hydrogen that's been lost, and therefore it's gained an electron. And reduction is gain. So if you think of oil rig, oxidation is loss, reduction is gain. So reduction is gaining an electron, and if you pick up the hydrogen, you are gaining an electron. So you can call it NADH or reduced NAD. And that stage 3, also releases 4 ATP molecules. But that means we have a net gain of 2 ATP from glycolysis, because we had to use 2 at the start. Take that away from the 4 that are made, we have a net gain of 2. So that's the stages written down. Let's have a look at it as a visual representation so it makes hopefully a bit more sense. So we start off with glucose, the 6-carbon compound. We then have that substrate level phosphorylation stage where we said we're going to be using 2 ATP molecules to add on 2 phosphate groups. So we phosphorylated it to form fructose 1,6 bisphosphate. That is now very reactive and unstable, so it splits straight away into 2 molecules of triose phosphate. That triose phosphate is then oxidized into 2 molecules of pyruvate, and it's oxidized by removing hydrogen, which is picked up by NAD to form reduced NAD or NADH, and in doing so, 2 ATP molecules are created. That happens on both of the triose phosphate molecules. So overall, the products we get from glycolysis are 2 3-carbon compounds of pyruvate, a net gain of 2 ATP, so although we made 4, we had to use 2, and 2 reduced NAD, or 2 NADH. And the pyruvate and the reduced NAD are going to be used in the later stages of aerobic respiration. So here we have our glycolysis happened in the cytoplasm. The next stages happen inside of the mitochondria. And as we just said, the pyruvate and the reduced NAD are going to be used in later stages. So those now need to be transported into the mitochondria, and they get transported in by active transport into the mitochondrial matrix. The next stage then is the link reaction. And this is when the pyruvate from glycolysis is oxidized to acetate. We have another NAD, which is going to pick up the hydrogen from this oxidation to create reduced NAD. And that's what we can see here. The pyruvate is oxidized into acetate, and we know it's oxidation because we have made a reduced NAD or NADH, which means the hydrogen must have been lost from pyruvate. You actually also have decarboxylation, which is the removal of a carbon in the form of carbon dioxide. So that's why we go from a 3-carbon compound pyruvate to a 2-carbon compound acetate. Now, acetate can't enter the next stage, which is the Krebs cycle, without the help of coenzyme A. So acetate then combines with coenzyme A to make acetyl-CoA or acetyl coenzyme A. And that is the link reaction. We don't have any ATP made in this stage, but we do have acetyl-CoA, carbon dioxide, and reduced NAD. And I've written 2 times for all of these because although we only get 1 NADH, 1 carbon dioxide, and 1 acetyl-CoA from the link reaction happens twice for every glucose molecule because we had 2 pyruvate molecules made in glycolysis. So the link reaction will happen twice for every glucose molecule. So that's why we've said 2 times those products. Next then is the Krebs cycle, which is still happening in the same location, which is the mitochondrial matrix. So here we can see the link reaction, and that's going to lead us into the Krebs cycle. And the first step of the Krebs cycle is oxaloacetate, which is a 4-carbon molecule, is going to combine with the acetyl-CoA. And because we've got a 4-carbon reacting with a 2-carbon, we get a 6-carbon molecule, which is citrate. And you do need to know the names of those compounds, oxaloacetate and citrate. At this point, the coenzyme A has served its purpose of helping the acetate react with the oxaloacetate, and therefore it's released and it goes to be reused again in the link reaction. We then have a series of redox reactions. There's actually lots of intermediates here, but you don't need to know any of those intermediates. You just need to know that there's a series of redox reactions, and that is because the carbon compounds are going to be oxidized, and these coenzymes are therefore going to be reduced. They're picking up the hydrogen that's being released from the carbon compound. So we end up making lots of reduced coenzymes. We have 2 carbon dioxides that are going to be lost, and that's how we go from a 6 carbon back to a 4 carbon molecule. And then we also have 1 molecule of ATP being produced at this stage. So from one round of the Krebs cycle, we get 3 reduced NAD, 1 reduced FAD, which is another coenzyme that picks up hydrogens, 1 ATP and 2 carbon dioxides. But much like with the link reaction, the Krebs cycle happens twice for every glucose molecule because we have 2 pyruvate made, therefore 2 acetyl-CoA, therefore the cycle happens twice. So for every glucose molecule, we have 6 reduced NAD, 2 reduced FAD, 2 ATP and 4 carbon dioxide molecules. So this is just splitting it up into those 2 stages, the link reaction and the Krebs cycle. So summary so far, we've gone through glycolysis, which happened in the cytoplasm. The pyruvate and the reduced NAD were actively transported into the mitochondrial matrix, which is where the link reaction and the Krebs cycle happened. So far, if we total up what we have produced in terms of coenzymes, because those are what are vital for this final stage, oxidative phosphorylation, we have made in total 10 reduced NAD from 1 glucose molecule and 2 reduced FAD from 1 glucose molecule. And those reduced coenzymes are really important for this final stage, which is oxidative phosphorylation. And this is where most ATP is created. So it involves an electron transfer chain, involves the movement of protons across the inner mitochondrial membrane or the cristae, and we get ATP being produced by ATP synthase. So let's go through what's happening then. In the mitochondrial matrix, that's where we've just had the link reaction and the Krebs cycle happening, producing lots of those reduced coenzymes. Those reduced coenzymes are now going to have the hydrogen removed, which we can see here, the hydrogen's been removed, and that hydrogen splits into the protons and electrons. And in this diagram, the electrons are represented by these star-shaped suns, and the protons are shown here. So the electrons are picked up by proteins embedded within the inner mitochondrial membrane. And this is the electron transfer chain, because the electrons then transfer along these proteins. As those electrons move from protein to protein, they release energy. And that energy is used to pump the protons from the matrix through these proteins into the intermembrane space. And therefore we end up with lots of those protons, which are hydrogen ions, all on one side of the membrane. And it's in the intermembrane space, which is the gap or the space between the inner membrane and the outer membrane. So we get this electrochemical gradient, which means we've got a lot of a charged molecule. So that's why it's electrochemical gradient on one side of the membrane. And that then results in the protons, these hydrogen ions, moving back across the membrane by facilitated diffusion down their concentration gradient. And the only protein they can move through due to complementary shapes fitting together is ATP synthase. And as those protons move down their concentration gradient back into the mitochondrial matrix through ATP synthase, that then causes the catalyzation of ADP plus PI into ATP. So as all of these protons move back down their electrochemical gradient, that's how we get lots of ATP being produced. Now, the final thing to be aware of is the role of oxygen here. And oxygen is known as the final electron acceptor. And that's because as these electrons move along the electron transfer chain or electron transport chain, at the end of the chain, if oxygen wasn't here to pick up those electrons, then they would stay in that protein, and therefore no more electrons can move along the chain. And if you don't have electrons moving along the chain, you don't have energy being released to pump the protons, you don't have an electrochemical gradient, and therefore you don't get ATP being produced. So it's really important that oxygen is this final electron acceptor, meaning it picks up those electrons so that the electron transfer chain can continue and we still get ATP being produced. And that oxygen picks up the electrons. It also picks up some of the protons, and that is how water is produced in aerobic respiration. So we then move on to anaerobic respiration, which is respiration in the absence of oxygen, and it only occurs in the cytoplasm of the cell. It involves only glycolysis from the stages we just saw, and the pyruvate that's made in glycolysis, in anaerobic respiration, it's reduced instead of oxidized. And in plants and microbes, it's reduced to form ethanol and carbon dioxide. In animals, it's reduced to form lactate or lactic acid. And this is done by the pyruvate gaining the hydrogen from the reduced NAD that were made in glycolysis. And the reason that needs to happen is that it's going to oxidize the reduced NAD back into NAD, and that means it can then be reused in glycolysis, so you can still at least have glycolysis occurring to produce 2 ATP molecules. So let's have a look at that represented visually. We said glycolysis occurs, so that's what we've already just gone through. And then this time the pyruvate is reduced. And we can tell it's reduction because that NADH becomes NAD, which is the oxidized version. And that hydrogen is picked up by, and that would be 2 NADH here, because it's going to be picked up by 1 each by these pyruvate molecules. And that is then going to reduce it into lactate or lactic acid. And that is actually toxic. Lactic acid is what causes muscle fatigue, but it is worth making that toxic molecule so that the NAD is regenerated and glycolysis can still occur to make at least some ATP. And anaerobic respiration can't happen indefinitely because that lactic acid would denature enzymes involved in this stage, and therefore it would then stop. And that's where we get this oxygen debt, where you need to take in lots and lots of deep breaths to get enough oxygen to fuse it into the bloodstream. And that oxygen can then break down the lactic acid in the liver back into non-harmful substances that can be used as respiratory substrates. Similar in plants and microbes, but instead of making lactic acid or lactate, we have ethanol and carbon dioxide being produced. So in terms of the efficiency of these reactions, one reduced NAD can result in a yield of 3 ATP molecules in that oxidative phosphorylation that we just saw, whereas one reduced FAD can result in a yield of 2 ATP molecules. And if we think back to the fact that we said from one glucose molecule, we create 10 reduced NAD and 2 reduced FAD, that results in 34 ATP molecules. Because we've got 3 times 10 and 2 times 2, so that's 34. But we also had a net gain of 2 in glycolysis and 2 produced in the Krebs cycle. So that's why overall from one glucose molecule you get 38 ATP. Whereas from anaerobic, we only get a net gain of 2 ATP in glycolysis. So there is a big difference there in how much ATP is produced in those 2 different reactions. So you do need to know this example as well, the rice plant adaptations, because rice plants have evolved several adaptations for growing in flooded or waterlogged conditions. So the erenchyma development, which is a specialised tissue with air-filled spaces in the roots and stems, this is to facilitate gas exchange, so oxygen and carbon dioxide fusing in and out between the roots and the surrounding water. It allows respiration when submerged by providing an oxygen pathway to the root tissues, and it reduces root weight, aiding buoyancy in those waterlogged conditions, so it will float. You also have ethanol fermentation in the roots, and that occurs under anaerobic conditions in waterlogged soils if there's limited oxygen. And that provides an alternative metabolic pathway for ATP production. That's going to help the rice plants to survive even in very, very low oxygen environments. And then lastly, we've got these resource allocations to the stems and the leaves. That's prioritising growth of aerial parts of the plant when it's submerged. And that is going to maintain photosynthesis and overall growth despite the adverse root conditions, and maximise exposure to sunlight and carbon dioxide for photosynthesis and carbohydrate production. So collectively, those adaptations enable rice plants to thrive in flooded or waterlogged environments, facilitating rice cultivation in high rainfall regions or irrigated paddy fields. So that is it for topic 12. I hope you found it helpful. If you did, look out for the later topics as well and check out my notes if you want even more support.